Dear students, bacteria are generally thought to be very destructive, disease causing, unuseful to our life. But bacteria, they are given to human welfare also. They are useful, they usefully contribute in many of the fields in our life. The first sector in which they contribute is in food production. Bacteria help raising the food's nutritive and calorific values. First of all, in the food production, we will study single cell proteins. Single cell proteins are actually the food supplements which are very rich in protein. The bacteria which are used to prepare single cell proteins have highest protein content in the range of 75% to 85% among the microorganisms. This capacity of bacteria, certain bacteria, is exploited to create single cell proteins as they could grow in simple and inexpensive nutrient medium. The single cell protein produced is also inexpensive. The bacteria which are generally utilized in preparation of single cell proteins is E. coli, Nocardia and Bacillus. These have very high protein content as I have mentioned right now. But there are certain limitations of single cell proteins because of their side effects. They induce formation of stones due to high nucleic acid and gout due to uric acid. They generally cause gastrointestinal disorders due to endotoxins produced by these bacteria. So they should be taken carefully because of their side effects. Now, many bacteria, they are used in supplying of food additives. Brevibacterium, for instance, produces monosodium glutamate, which enhances the flavor of the food, and it is widely used in the Chinese food which we see and which we eat in the hotels and restaurant. This is normally called as Ajinomoto, and it's an necessary necessary thing in the kitchen these days. Bacillus subtilis is used in production of amylase, and this amylase is used in formation of syrups. Bacillus species used to produce glucose isomerase and protease, which are used for sweetening of syrup and tendering of milk respectively. Corynebacterium produces lysine, which is also used in industries. Enterobacter produces diacetyl reductase, which removes off flavor of the beer produced and fruit juices which are produced in the factories. Mycobacterium lactum produces vitamins. Micrococcus luteus and rosens helps in meat currying. The leuconostoc mesenteroids produces high sugar concentrations in the syrups, and it is widely used in ice creams, in making of ice creams. Leuconostoc cramoris ferments the citric acid of milk and also stimulate lactic streptococci, so-called lactic starter for butter, milk and cheese. Then we have Propionibacterium species which ferments lactate and contributes to enhancement of flavor of cheese. Staphylococcus thermophilus used in cheese making. We have Staphylococcus lacti, which is a very important member of dairy industry. Then we have Bacteria, which indicates the pollutants at the site of their at the site where the pollutants are present. Bacteria are highly sensitive to environment fluctuations. Therefore, the growth limiting factor of bacteria, if present in a pollutant, we can easily determine that pollutant is present by determining the growth patterns of the bacterial colonies. Therefore, the bacteria, they are used as indices for feeling the pulse of the system, and they serve as bioindicators as far as pollution is concerned. Bacteria, they are play a very important role in recycling of minerals, for instance in carbon, nitrogen, oxygen, sulfur, phosphorus, etc. Photosynthetic and chemosynthetic bacteria converts carbon dioxide into organic carbon through fixation process. Methanogenic archaebacteria generates methane from CO2 and H2. This is known to you. And bacteria, they play a very important role in bioremediation. Bioremediation is actually the removal of pollutant from a specific site or reclamation of the land or water body to its original form. In bioremediation procedure, we use genetically engineered bacteria such as Pseudomonas fetida, which has four plasmids artificially introduced inside it. One is OCT, which degrades hexane, octane, and decane at the polluted site. Then we have XYL, which degrades xylene and toluene. We have CAM plasmid, which degenerates camphor. And finally, the fourth one is NaH, which degrades naphthalene. That means wherever you find these pollutants, and if you introduce Pseudomonas fetida formulations there, the Pseudomonas fetida will degrade all these pollutants and will reclaim the site. Now we have oil eating microbes, which are also called as superbugs, which are patented by a company Exxon, which cleans up the spills in oceans and water bodies. Another name for these oil-eating microbes is oil zappers and oleovorous, as prepared by Tata Institute of prepared by Terry. These oil zappers and oleovorous, as they reclaim the oil spills area. This Terry is Tata Engineering Research Institute. Now bacteria also find their way into agriculture field. They convert highly lignified straw and bagasse into biofertilizers, which is also called as organic compost. The symbiotic or free living bacteria, they converts molecular nitrogen, which cannot be used normally, to usable forms like ammonia. Then we have symbiotic nitrogen fixers like rhizobium, azorhizobium, mesorhizobium, cynorhizobium, and bradyrhizobium, which fixes the atmospheric free nitrogen into the usable compounds. Among the free living nitrogen fixtures, we have Azotobacter and Azospirillum. They tightly binds to the roots of non-leguminous plants like corn and enable them to fix nitrogen by converting molecular nitrogen to assimilable ammonia. This is very important from exam's point of view. What is the methodology followed by nitrogen fixers. The nitrifying bacteria converts ammonia to nitrate by aerobic oxidation. Such bacteria, nitrifying bacteria, are Nitrosomonas, Nitrosococcus, and we have Klebsiella pneumoniae, which has 17 gene in place for nitrogen fixation in a single plasmid. Bacteria, they are also involved in biological control of some pests or insects or etc. For instance, Bacillus thuringiensis, a bacteria which produces toxic crystals which kills the gypsy moth larvae, which causes great economic loss in the crop of cabbage. Bacteria, they are widely used in biotechnology and in microbiology. They are even more widely used in pharmaceutics for production of antibiotics, vitamin B2, vitamin B12, and vitamin C. They are also involved in formation of insulin, somatostatin, cortisone, and human growth hormone, which are produced artificially or which are produced through biotechnology, in fact. And also they are involved in making of vaccine by pure viral antigenic determinants. They are involved in formation of interferons. Also interferons actually are antiviral agents which are produced by the body against the viral infection. In agriculture, NIF genes, which is a nitrogen fixing gene, is transferred from nitrogen fixers like Klebsiella pneumoniae to yeast and plants so that they can make or fix their own nitrogen into the compounds which can be used for the purpose of assimilation. Cold tolerant gene transferred from Arctic fish to tomato so that tomato may not die of chill or cold. This is also achieved through bacteria because the plasmid, they serves as cloning vehicle. The area where oil gets spilled, reclaimed through superbugs, oil zappers, oleovorous, as I have told you in the previous slide. Now, bacteria is also widely used in industries, fermentation industries, and they maximize production by employing the industry. They maximize the production by employing high yielding strains which are capable of utilizing very low cost and inexpensive raw material, and the production is raised many folds. For example, the production of penicillin is raised 10,000 fold higher than the Fleming's original method. Then fermentation industry. Fermentation is actually the energy yielding process in which an organic compound is converted into lactic acid. For example, glucose gets converted into ethyl alcohol, and this is done anaerobically. And through fermentation procedures, we make vinegar, wine, and lactic acid. I am going to mention the formation of vinegar and lactic acid in this slide. The substrate which are used for formation of vinegar through fermentation is malt, wort, grape juice, apple juice, and cane juice. Now, these substrates, they are acted upon by the bacteria which is called as Acetobacter courliensis. They are also termed as inoculants in the industry parlance. The procedure which is followed is trickling process, and from the top liquor is distributed over the packings which provides or which helps in growth of Acetobacter. This liquor slowly gets acidified as it trickles down at the bottom. This process is repeated many a times. And this is how the process of acidification is achieved, and this is which is an exothermic process. Now tower is used as chimney and cool air is drawn at the base so as to uh, reduce the temperature of the system. Another product which is widely made in the fermentation industry is formation of lactic acid, which is a colorless, odorless, and is used in the food industry as an acidulant in confectionaries and in fruit juice industries. Also used for curing of meat and fish. The substrate which is used for this lactic acid fermentation are carbohydrates. In carbohydrates, we use corn, potato, and starch, also molasses and whey. Inoculants used here is lactobacillus. We prepare an incubation tank which is maintained at around 40 degrees centigrades and filled with pasteurized whey along with starter culture and calcium hydroxide to neutralize the acid and to promote yielding of calcium as acetate. This is then boiled and purified, and later and lastly the lactic acid is recovered from the incubation tank. So students, this is all in the topic entitled Bacteria for Human Welfare. Thank you very much.